Vamos a empezar con la primera plática del segundo bloque, con un tema de bioingeniería. Tenemos con nosotros al doctor Pascal Jaouen. El doctor Jaouen es doctor en bioingeniería, bioprocesos, dirige la carrera de bioingeniería en Nantes Polytech, la universidad que acaba de firmar el convenio de colaboración con la Universidad Autónoma de Querétaro y con la cual va a haber intercambios de estudiantes y programas conjuntos de investigación. Además de ser director de esa carrera, el doctor Pascal Jaouen dirige un laboratorio que se llama GPA y que es un centro de investigación y transferencia tecnológica. En este laboratorio trabajan 500 investigadores, trabajan de manera específica sobre el tema de las microalgas y es uno de los laboratorios más destacados en este tema a nivel mundial. Además de haber publicado en revistas científicas más de 200 artículos, el doctor Jaouen también es socio de una startup que se llama Algae y que se dedica a producir componentes derivados del cultivo de las algas. Entonces, les pido por favor un fuerte aplauso para el doctor Pascal Jaouen.
Okay. Thank you very much. Thank you very much for your kind invitation. And it is, of course, a pleasure for me to discover your activities. And as you said, Erwan, my talk will focus on microalgae, photobioreactors, biofuels, and the use of microalgae for the ingredients and also in food and feed and also cosmetics. As you said, I am integrated and I am professor of University of Nantes and head of a large research institute of some 450 researchers and also director of a school of engineering, a graduate school of engineering, with the Department of Process and Bioengineering in Nantes. So it is located in Western France. So we are located here in Western Europe. And first of all, some dates about the University of Nantes. It is 45,000 students, 20 faculties, and 20 faculties and institute, and one graduate school of engineering. As a whole, 70 research units. My presentation is divided in 3 parts. The first part is dealing with research mainly. The second concerns the training aspects. And the third one is more oriented towards industry. First of all, research on photobiotechnologies, microalgae and cyanobacteria. My own lab, GPA, is a research unit associated to the French National Research Center, CNRS. It is a rather well-evaluated lab, A+, which is very important in our French system. We are very proud of that. GPA has 45 researchers working specifically on microalgae and process to produce and valorize compounds contained in the microalgae. So we are focused on photobioreactors and separative techniques for the production of microalgae for food, feed, cosmetics, aquaculture, and I have also used consultant for the European Space Agency for the reuse of water and production of food in a very special environment. We have also about several PhD students on bioenergy, biofuels, and biomaterials. So we are located here in Nantes, Saint-Nazaire, very close to the famous resort station La Baule in south of Brittany. So we are interested in photosynthetic microorganisms. As you probably know, they have different sizes, shapes, and above all, cellular contents. The principle is the assimilation of the inorganic carbon with the presence of light, water, and specific nutrients to transform it as an organic matter. Our scientific approach is to investigate the bottlenecks that limit the productivity of photobioreactors. Photobioreactors are really the heart of the process. Then we have a modeling approach of the more influent parameters, and then we can transpose to practical cases for optimization. So we conduct experiments in lab scale in fully controlled photobioreactors. It is typically an approach of chemical engineering type. So first example, the engineering of photobioreactors. We develop several scientific problematics. The design and the development of new geometries, both for solar and artificial light. Studies on the optimization of the photobioreactor, for example, for high cell density. What's the... What's that? Photobioreactors for high cell density. And also, it is very important for engineers to have a modeling approach. We study the kinetics. The modeling is very important for the scaling up and for control. We have studies on metabolism, flux analysis, and physiological induction, which is very important when we want to orient the production of the cell to certain metabolites of interest. Next slide, please. Let's try with this one. It's good? Okay, let's continue. As I said before, we have strong studies on photobioreactor modeling. As I said before, the first part of my presentation is more scientific, what we do, and then we have more practical examples. So we conduct experiments in fully controlled conditions. It is very important that because some of you that work on this kind of biomass know probably that one of the main problems with microalgae is the possible contamination with other species that are not desired in the system. So experiments are conducted in fully controlled conditions. So kinetics model, scaling up and control, and the specific study on radiative transfer modeling. We are very interested on photobioreactor optimization, and particularly concerning hydrodynamics, because it is very important both for transfer, for radiative transfer, but also for mass transfer between the cell and the medium which contains the nutriments, and there are, of course, many exchanges between the cell and the medium. So the hydrodynamics is one of the issue in such reactors. We study, for example, very specific swirling flows to avoid the development of biofilm that limit the productivity at the interface photobioreactor light. So this is an example of the very, very classical raceway. Concerning photobioreactors engineering, as I said before, we try to develop a small-scale system, high-volumic productive photobioreactors, closed raceways technology in order to study the CO2 transfer, which is an issue for an industrial application of microalgae. And above all, what is very important for us, and this system is patented, a solar thin film, whose name is Algae film, because the productivity that are obtained in this kind of tool are much higher than those obtained with other types of systems. So we have we are very enthusiastic about this thin film photobioreactor. One of the main, we have about 20 PhD students on all aspects of photobiotechnology, from metabolic engineering to fluid dynamics to water treatment and recycling, extraction, with all the activities that we generally find when we work in process engineering. So the production of lipids, and particularly TAG, triacylglycerides, is very important for the production of biofuels. And there, after the strain is correctly selected, and we have to optimize the experimental protocols, then we have to develop predictive models for the productivity, and also to work on the scaling up for high volume production. Another part of also concerning energy, we have also developed two theses on the hydrogen photobioproduction, and here this is our scientific approach to investigate the biological pathways involved during the H2 production, with an analysis of metabolic flux and investigation of new protocols towards large-scale production. Of course, in a research lab, we are involved in many programs, both national programs supported in France by the French ANR, Association Nationale de la Recherche, and also European programs. All the programs are dealing with aquaculture, with the development of specific photobioreactors for the aquaculture, reactors for the production of lipids and exopolysaccharides that are, of course, of interest in many fields, POH2 production, and solar production. A very promising concept concerns what we call algae refinery or biorefinery for the production of specific metabolites of interest. So this is a representation of what is the biorefinery concept applied here in the case of one specific microalgae, the case of Porphyridium cruentum. As you see, following different treatment technology bricks that are linked one to another, it is possible to extract selectively each component of a biomass without denaturing the others. It is really a challenge, and it is, to our point of view, very, very innovative. It is exactly that has been developed more than 70 years ago in petroleum industry and more recently in the milk industry. We want to go to a kind of cracking, a very fine separation of each component of the microalgae. And for example, concerning the Porphyridium cruentum, it is possible to extract. We have shown in a recent thesis that it is possible to extract colorants, phycoerythrin, which is a protein used as an immunofluorescent probe, and which also present a good potential for food coloring as food coloring substance, can be separated without damaging the polyunsaturated fatty acids for potential use as nutritional supplements. We can also extract zeaxanthin, which is with its antioxidative activity, so as the superoxide dismutase, and also the exopolysaccharides. So it is really the great challenge now to develop global economic models interesting for the biomass production and valorization with an economic positive balance. This is a very important project we have on the site of Nantes-Saint-Nazaire. The name of the project is Défi Microalgues. It is a kind of research and development platform dedicated to the semi-industrial production of microalgae using all the different technologies available today, and to develop more specific technologies that are not used as of today, and to compare the performances of each technology for each objective. In the underground of the platform, we have a downstream processing platform specifically dedicated to the biorefinery. It is a sensible part of the project to harvest, to produce, to harvest, and then to extract the different metabolites of the microalgae. And of course, we will have with this extraordinary tool the possibility to work on the problematic of scaling up. We can produce microalgae at very small scale. It is very more difficult to transpose to large or very large scale. So it is really a very large project. It will probably be one of the most well-equipped platform of this type in Europe. Very original demonstrator. We have invested 4 million euros on this platform just for the infrastructure, and we will add 5 other million euros for the specific equipment and to develop research and development programs. The second part of the presentation, much shorter, concerns training. As you have seen this morning, we have now a cooperation agreement between the University Autonome of Querétaro and the University of Nantes. This master degree can be the framework of this collaboration, and with possible mobility of students from Nantes to Querétaro, and also from Querétaro to Nantes. We have modules relevant to bioprocess, methodology tools for the engineer, and microbiological and enzymatic engineering, economic development of biotechnology of microalgae, and so on, food engineering applied to sea products. So we have this master exists for 8 years, and we try to develop, to make it as to give it a new orientation and to transform it as an international master of excellence. This is an example of our students in Nantes Polytech, the ceremony for the diplomas. Okay, so let's move to the third and the last part of the presentation. As we said just before, we are very interested to reinforce what we call a virtuous cycle, gathering together research, training, and innovation linked to the industry. And it is the third part of the presentation, which concerns our implication in a small SME, the AlgoSource enterprise, divided in 2 parts. We have this. I am a partner, scientific partner. I'm a shareholder and scientific counselor to this enterprise. We have AlgoSource products with the objective to product and some functional ingredients from microalgae and cyanobacteria, nutraceutical and cosmetic industries. And more recently, we have created AlgoSource technologies, which is more focused on engineering and process aspects for the production of and valorization of microalgae. So the different species, some people in here know very well, some of them, Arthrospira, spirulina, very well known in Mexico, of course. Senedesmus, Phaeodactylum nanochloropsis for the production of specific lipids, Porphyridium with its polysaccharides and superoxide dismutase, and also Hematococcus for the polyunsaturated fatty acids. So these are the activities of the AlgoSource group. Alpha Biotech is more focused on the production and also algo refining. We have started this concept, the establishment of this concept at an industrial scale with spirulina. After harvesting, the microalgae, the biomass is then extracted from specific exometabolites. Then, after disruption, we can extract the phycocyanin, which is a high-value product, and then lipid extraction allows us to obtain microalgae oil and different other additional treatment lead to structural molecules of interest. The final residue is also used industrially. Concerning the AlgoSource technologies, it is a very small enterprise, 6 partners, 4 employees, but it is a fast-growing SME. And our core competency is the process integration, and we have 4 main topics. The first topic concerns, it's very, very important for for industries producing CO2. The first topic is the biosequestration of CO2, the recovery of heat, and the wastewater recycling, particularly nitrogen and phosphate. Then the second topic deals with the mass, which is more classical, the mass production of microalgae. The third topic is on the harvesting of microalgae biomass. Harvesting techniques can be very different from one microalgae to another, since their size and their shape are very different. And the differentiation of volume massic is between the cell and water can slower the separation. And at least the fourth topic, which is refining, it is a more strategic topic. It concerns the refining of the biomass itself with application in food, oils, vitamins, fatty acids, for feed. But feed not only proteins, polysaccharides can also be interested, polyunsaturated fatty acid also. And we can also give for the chemistry green building blocks, starch, sugars, acids. Biofuels is also interesting for the production of diesel, kerosanol, and also biogas. So in this small spin-off of my lab, we work essentially with French and European programs, and we are coordinator or partners, simple partner of some large European programs. Okay, thank you for your attention. This is the place where we are located. It's a nice place, very close to the Loire estuary. Nantes is over there, and here is the city of Saint-Nazaire.
Thank you very much, Pascal. This is a present from Querétaro University and from the cluster. Could you tell us, Pascal, when is going to start the next generation for the master? If some students here are interested for going to this nice place, when can they leave from Querétaro?
We've just discussed this topic this morning with your colleagues from the Secretariat of the Director. We think it is possible to begin concretely, really the collaboration from September. During the first semester, courses will be given in the site of Saint-Nazaire, and then the second part of the year, students have to compete training in industry or in a research lab, even in France or Mexico if possible.
How many students from different countries can you receive in your Master International?
Usually the population is limited to 15 to 20 students, but the best students speaking a bit French and also a bit English with a good courses will be favorable examined and possibly integrate in our group.
Okay.